Welcome back, everybody, to the Deep Dive. We're diving into something truly mind-blowing. Today, it's all about partial cellular reprogramming.
Oh, yeah.
This is basically like hitting the pause button or even rewind on aging, but at the cellular level.
That's a good way to put it.
Please don't forget to like the video and subscribe if you want more content like this. So we're not talking about turning back the clock completely and turning a skin cell into a stem cell.
Right.
This is more nuanced, right?
Yeah.
We're talking about nudging cells to a slightly younger state.
Exactly. Kind of like a cellular tune-up, not a complete overhaul.
So where are we getting all this cutting-edge info from? Well, we're basing our deep dive today on a super comprehensive review, hot off the press, in Aging Research Reviews, published just this year.
That's right.
So our mission, should we choose to accept it, is to figure out what actually changes in these cells during this partial reprogramming.
Yes.
And, of course, what this could mean for understanding how we age, and maybe, just maybe, finding new ways to slow it down or even reverse it.
Absolutely.
So, first things first, what exactly are we talking about when we say partial reprogramming?
Well, imagine you have a mature, specialized cell, like a skin cell or a muscle cell.
Okay, got it.
In partial reprogramming, we expose that cell to factors that can make it more versatile, kind of like a stem cell.
Okay.
These are often called pluripotency factors, and they can guide a cell to become different types of cells.
Ah, like a cellular toolbox.
Right, but here's the key. We do it in a very controlled and limited way. The cell gets just a taste of these pluripotency factors, not the full course.
So it's like giving it a little nudge in the right direction.
Yeah, we're not trying to erase its entire identity and turn it into a blank slate stem cell, which is what happens in full reprogramming.
Makes sense. So it's like we're tapping into its potential, but not letting it go wild, right?
Exactly. We're gently coaxing it to a slightly younger, more adaptable state.
Okay, so how do they actually do this in the lab?
Well, there are different methods. One common approach uses a specific set of pluripotency factors called the Yamanaka factors.
I've heard of those, yeah.
They might introduce these proteins for a short period, like a few days, or in brief pulses, just enough to trigger the reprogramming process without pushing it too far.
Interesting. So it's like a little cellular boot camp.
I like that. And what's amazing is that this has been done not only in cells grown in a dish, but also in living mice.
Wow. In living animals. That's pretty impressive.
It is, and it opens up some really exciting possibilities for future therapies.
So what's happening inside these cells when they're exposed to these reprogramming factors, even for a short time?
One of the most consistent changes happens at the level of the cell's DNA packaging, the chromatin.
Okay, remind us what chromatin is again.
Sure. It's the way DNA is organized within the nucleus. You can picture it like a super long thread that needs to be neatly packed into a tiny space.
Okay.
Well, during partial reprogramming, there's a lot of dynamic remodeling of this chromatin structure.
Like rearranging the furniture to make the space more efficient.
Kind of. Specifically, there are shifts in something called histone modifications. Histones are proteins that DNA wraps around, and these modifications act like switches and dials, controlling how accessible genes are.
Ah, so it's like adjusting the volume on certain genes, right?
Exactly. And partial reprogramming tends to make genes associated with a younger, more adaptable state more accessible, while making those that define the cell's specific job less so.
So it's kind of like giving the cell a chance to explore its options again.
That's a great way to put it. It's like reminding it of its hidden potential.
Fascinating. Are there other major changes happening inside these partially reprogrammed cells?
Absolutely. Beyond the changes in gene regulation, there's a clear impact on how cells respond to stress.
Stress is a big factor in aging, isn't it?
It is, and as we age, our cells become less able to cope with stress.
Right.
Well, one of the hallmarks of aging is chronic inflammation, which contributes to many age-related diseases.
Yeah, we hear a lot about inflammation these days.
And for good reason. But what's encouraging is that partial reprogramming seems to generally reduce inflammation.
That's good news.
It is. We also see an improvement in autophagy, which is the cell's internal recycling system. It's kind of like the garbage disposal and recycling center of the cell.
And that tends to slow down as we age, right?
Exactly. With age, cellular waste products start to build up, but partial reprogramming seems to give autophagy a boost.
Okay, so that's two major stress response systems that seem to benefit. What about cellular senescence?
That's a bit more complicated. Cellular senescence is when cells basically enter a retirement phase and stop dividing.
And sometimes they can cause trouble.
They can. Senescent cells can release harmful molecules that damage surrounding tissues and contribute to aging.
Right.
The effect of partial reprogramming on senescence is a bit mixed. Some studies show a reduction in markers of senescence, which is a good sign, but it's not always consistent. And it seems to depend on a lot of factors, like the cell type, the reprogramming method, and even the species.
So more research needed on that front.
Definitely. We still have a lot to learn about how senescence is affected by partial reprogramming.
What about the cell's powerhouses, the mitochondria?
Good question. Mitochondrial function is crucial for energy production, and it tends to decline as we age.
Leading to fatigue and all sorts of problems, right?
Right. But what's really exciting is that partial reprogramming often revitalizes mitochondria.
Really? That's great news.
It is. We frequently see an increase in something called oxidative phosphorylation, which is the main way cells generate energy using oxygen.
So it's like giving those mitochondria much-needed tune-up.
Exactly. And finally, there's the extracellular matrix, the structural support network surrounding cells, which is also affected.
Okay, we haven't talked about that yet.
Right. But the changes here can be quite varied, and we need more research to fully understand how the extracellular matrix is impacted by partial reprogramming.
So it's a complex picture with a lot of moving parts.
It definitely is.
Now, you mentioned earlier that reprogramming is more like a spectrum, not a clear-cut set of stages. Can you unpack that for us?
Yes, it's not as simple as flipping a switch. It's a gradual process with cells moving along a gradient of change.
Okay.
But to help make sense of it, scientists have proposed models with different phases. One popular model describes 3 main phases: initiation, maturation, and stabilization.
Walk us through those, please.
Sure. In the initiation phase, the cell starts to suppress the genes that define its specialized function, and it starts to activate genes associated with a more flexible, stem cell-like state.
Okay, so it's like loosening up its identity a bit.
Exactly. A key process during this phase is often something called mesenchymal to epithelial transition, or MET-T.
Whoa, say that again?
Mesenchymal to epithelial transition, or MET-T.
Yeah.
Basically, it's like the cell is shedding some of its migratory, connective tissue-like characteristics, and it's becoming more organized and structured.
Okay, got it. So it's becoming more epithelial.
Exactly. Then comes the maturation phase, where we start to see the expression of some actual pluripotency genes.
The genes that give stem cells their special abilities.
That's right. But remember, in partial reprogramming, we're usually stopping before we reach the final phase, which is stabilization.
Where the cell becomes a full-blown, induced pluripotent stem cell.
Yes. We don't want that to happen.
Too risky.
Exactly. We're aiming for that sweet spot in the middle, where we get some of the benefits of rejuvenation without the risk of uncontrolled cell growth.
Makes sense. So you're carefully controlling how far you push the reprogramming process.
Precisely. And what's really fascinating is that in the early stages of reprogramming, we often see a downregulation of EMT.
Which is the opposite of MET-T.
Exactly. It's like reinforcing that shift towards a more epithelial-like state.
Okay, so the Yamanaka factors are playing a role in directing this transition.
Yes. They actually directly suppress factors that promote EMT and activate genes associated with epithelial traits.
Wow, so they're like the conductors of this cellular orchestra.
That's a great analogy. They're orchestrating these complex changes in gene expression.
And what happens when you remove the reprogramming factors? Do the cells stay in this more youthful state?
Well, that's another interesting aspect. In many cases, when partially reprogrammed cells are allowed to recover, they tend to revert back towards their original state.
Oh, so it's not a permanent change.
Not always. It seems there's some kind of cellular memory that pulls them back to their original identity.
Like a rubber band snapping back to its original shape.
Yes, exactly. But even though they might revert back, they seem to retain some of the benefits of reprogramming, at least for a while.
That's promising.
Yeah.
So let's talk about what's happening at the level of the epigenome, which you mentioned earlier. What's going on with that cellular memory?
The epigenome is like a layer of instructions that sits on top of our DNA, influencing which genes are turned on or off.
Okay, I'm following.
Well, during reprogramming, both full and partial, there are massive changes in the epigenome.
So it's like rewriting the cell's instruction manual to some extent.
In a way, yes. It's like erasing some of the marks that have accumulated over time and adding new ones that promote a younger, more versatile state.
Interesting. So what kind of specific epigenetic changes are happening?
One early change is something called epigenetic priming, where specific histone modifications appear at key pluripotency and ME genes very early in the process.
Even before those genes are fully activated.
Exactly. It's like putting down the groundwork for the reprogramming machinery to move in.
Okay, I'm picturing a construction crew setting up scaffolding before they start building.
That's a good analogy. Another important step is removing repressive marks, like H3K27Me3, which are taken off by an enzyme called JMJD3.
Those repressive marks are like roadblocks preventing access to certain genes.
Exactly. And JMJD3 comes in and clears the way. Interestingly, vitamin C can actually enhance JMJD3's activity.
Wow, so something as simple as vitamin C can influence reprogramming.
It seems so. It highlights how interconnected these molecular pathways are.
Amazing. Are there other epigenetic hurdles that reprogramming factors need to overcome?
Yes. Another one is heterochromatin, which are tightly packed regions of DNA that are generally inaccessible.
Like a locked vault.
Yes, and these regions often contain pluripotency genes that need to be unlocked during reprogramming. Scientists have found that reducing the activity of enzymes that maintain heterochromatin can speed up the process.
So it's like finding the key to the vault.
Exactly. As reprogramming progresses, we see the accumulation of active marks like H3K36Me3 on the genes that are being turned on.
It's like adding signposts, making it easier for the cellular machinery to find those genes.
That's a good way to think about it. At the same time, we see the decrease in the activity of enhancers that control the expression of genes related to the cell's original identity.
So it's like turning down the volume on those genes and turning up the volume on the pluripotency gene.
Precisely. And two of the Yamanaka factors, OCT4 and SOX2, play a special role here. They're like pioneers, able to bind to and open up those previously inaccessible regions of chromatin.
They're paving the way for other factors to come in and activate those genes.
Exactly. It's a remarkable process, and we're only just beginning to understand the intricate details.
This is mind-blowing stuff. Are there any differences in how this unfolds in different species?
Yes, there are some interesting species-specific variations. While the general principles are conserved, the specific genes targeted by the Yamanaka factors can differ between mice and humans.
Okay, so there are subtle differences in the choreography.
Exactly. And it also depends on the environment surrounding the cells. In a living organism, the surrounding tissue and the extracellular matrix can influence how cells respond to reprogramming.
So it's not just about the cell in isolation. It's about its interactions with its neighbors.
Absolutely.
Now, with all these positive changes happening inside cells, there must be some potential downsides.
Of course, there's always a balance, and one of the biggest concerns is the potential for cancer.
That's a big one.
It is. Because we're essentially reactivating genes that are normally active in very early development, there's a risk that cells could start dividing uncontrollably.
So how do you minimize that risk?
That's a major focus of research. One approach is to carefully control the duration and intensity of the reprogramming treatment.
Like finding the sweet spot between rejuvenation and safety.
Exactly. And scientists are also exploring alternative reprogramming cocktails that don't include certain factors, like MYC, which is a known oncogene.
So, removing the potentially dangerous ingredients.
That's the idea. There's also a concern that reprogramming might inadvertently activate precancerous cells, making them more likely to become full-blown tumors.
Oh, that's worrying.
It is. And it's something that needs to be carefully monitored in any future clinical trials.
And what about senescent cells? Can they be a problem?
Well, as we discussed, senescent cells can sometimes re-enter the cell cycle after reprogramming, which could also contribute to uncontrolled growth.
So it's like waking up a sleeping giant.
In a way, yes. But there's also evidence that some reprogramming methods can actually reduce the number of senescent cells, so the effects are complex and context-dependent.
It's a delicate balancing act for sure.
It is. We need to proceed cautiously and do our homework to ensure that any future therapies are both effective and safe.
Now, are all cells equally susceptible to reprogramming?
That's a good question, and the answer is no. Some cell types are more readily reprogrammed than others.
Why is that?
It could be due to differences in their epigenetic landscape, their ability to take up reprogramming factors, or even their inherent regenerative capacity.
So some cells are naturally more adaptable than others.
That seems to be the case.
Let's shift gears now and talk about the exciting potential benefits. What impact is partial reprogramming having on those aging clocks we hear so much about these days?
This is where things get really interesting. Both partial and full reprogramming have shown the ability to rewind those aging clocks, at least to some extent.
Wow, really turning back time.
In a way, yes. Studies have shown that in living mice, treating them with Yamanaka factors can actually reduce their biological age as measured by these clocks.
That's amazing.
Yeah.
And is it just a temporary effect, or does it last?
In some cases, the reduction in biological age persists even after the treatment is stopped.
That's encouraging. So it's not just a cosmetic change. It's influencing the cell's underlying programming.
It seems so. But we still need to figure out exactly what these clocks are measuring and how accurately they reflect true biological age.
There's always more to learn, right?
Absolutely. But the fact that we're seeing these consistent changes across different cell types and tissues is very promising.
It is. What about more visible signs of rejuvenation?
Well, we're also seeing encouraging results with more direct measures of rejuvenation. For example, in the brain, partial reprogramming has been shown to increase the production of new neurons, which is essential for learning and memory.
So like boosting brain power.
Exactly. And we're also seeing improvements in other tissues, like the liver, the eyes, muscles, skin, and even the heart.
It's like a whole body tune-up.
That's a great way to put it. It seems that partial reprogramming can activate regenerative processes in a variety of tissues.
This is truly groundbreaking stuff. And what about those stress responses we talked about earlier? Is there evidence that partial reprogramming can improve them at a functional level?
Yes, there is. We're seeing a general trend of decreased inflammation in response to partial reprogramming.
That's great news.
It is. And we're also seeing improvements in autophagy, suggesting that cells are better able to clear out damaged components.
That's like giving the cells a spring cleaning.
Exactly. And although the effects on cellular senescence are more complex, some studies do show a reduction in the number of senescent cells and their harmful secretions.
So it's like tackling multiple hallmarks of aging at once.
Precisely.
Now, are there any surprising similarities between partial reprogramming and the aging process itself?
That's an interesting question, and it might seem counterintuitive, but there are some intriguing parallels.
Really? How so?
For example, both aging and partial reprogramming can sometimes lead to increased inflammation in the brain.
That's unexpected.
It is. And we also see some overlap in the genes that are upregulated in both processes.
So there might be some shared pathways involved.
It's possible. And it highlights the need to carefully study both aging and reprogramming to fully understand how they interact.
And with all this exciting research happening, how close are we to seeing partial reprogramming used in the clinic to treat human diseases?
Well, there's still a lot of work to do, but the potential is huge. One promising area is in cancer treatment, where reprogramming could potentially be used to correct the epigenetic changes that drive tumor growth.
That's a bold idea.
It is. And there's also growing interest in using partial reprogramming to treat age-related eye diseases.
Because the eye is a more contained environment, right?
Exactly. And it's easier to monitor for potential side effects.
Makes sense. So we're still in the early stages, but the future looks bright for this field.
I agree.
Before we wrap up, are there any limitations to the review we've been discussing that we should be aware of?
Yes, it's always important to consider the limitations of any scientific study or review. This particular review focused primarily on studies that combined both omics data and phenotypic data.
Meaning, they looked at both changes in gene expression and observable changes in cells or organisms.
Exactly. And that might have excluded some earlier studies that could provide valuable insights into the mechanisms of reprogramming.
Okay, so there might be some missing pieces of the puzzle.
That's possible. Also, there was a lot of variability in the experimental designs of the studies included, which can make it difficult to compare results directly.
So, more standardized approaches are needed.
Definitely. And finally, it's crucial to distinguish between the cells that are actively being reprogrammed and those that have reverted back to their original state.
Because the changes happening in those two states can be very different.
Exactly. More research is needed to understand the dynamic nature of these changes over time.
Well, this has been a truly fascinating and enlightening exploration of partial cellular reprogramming. What are some of the key takeaways for our listeners?
I'd say the main takeaway is that partial reprogramming can have a profound impact on fundamental aging processes.
By influencing gene expression, stress responses, and even metabolism.
Exactly. And the fact that we're seeing these effects across different species and tissues is very encouraging.
It really does suggest that we might be on the verge of a revolution in how we approach aging and age-related diseases.
I'll agree.
But of course, there are still challenges to overcome, particularly regarding safety.
Absolutely. We need to proceed with caution and do more research to ensure that any future therapies are both effective and safe.
So as we reach the end of this deep dive, What are some questions for our viewers to ponder?
Well, given the complexity of this field, I think one question is, what's the most promising avenue for future research?
Should we focus on understanding the basic mechanisms, developing safer techniques, or prioritizing specific applications?
It's a tough question, but a crucial one.
And on that note, we'd love to hear your thoughts in the comments below.
Please share your insights and let us know what you find most exciting about this revolutionary field.
And of course, don't forget to like the video and subscribe if you want more deep dives into cutting-edge science.
We'll see you next time.